In phylogenetic analysis, we want to find or infer relationship between different species. Now, how we can find this relationship? If we say that from one species to the other species and from the second to the third species, there is no constant time. That, for example, from one species to the second species, it takes, suppose, say 300 million years, and from second to third, it takes only 2 million years. So, can we infer relationship if this is the scenario? Answer is no. What we say that basically information or relationship between different species and their evaluation time remains constant. So that is basically background for a concept called molecular clock. So what is molecular clock? आइए देखते हैं. Basically, molecular divergence means that change in information is correlated with correlation of time. So what happens? Change in molecular information, which is our DNA, RNA or protein structures, it is correlated with change in time also. So what happens in DNA or other macromolecules, it is also constant across time also. This molecular clock has certain other names such as evolutionary clock, gene clock, species clock, and then how it is used and why it is used. Basically, it gives us information about the occurrence of different speciation events or radiation events. So those events are basically related to when one species corresponds or converts into the other species. So if I move on, there are two parts again, speciation or radiation. Speciation is directly related to change in species information, and then radiation may be from radioactive information. So what happens then? What is the usefulness? So using this molecular clock, using fossil constraints or fossil information, we can deduce time of change from one species or from one organism to the other organism. There are certain hypotheses which are necessary for molecular clock information. So what are those hypotheses? For example, we say that if protein sequences evolve at constant rates, then they can be used to estimate the time that caused those sequences to diverge. And the other thing is, this thing is similar to information change during the geological information. So what happens is that if we know that among two species or more than two species, there is a constant change in their protein structure, then we can diverge or we can find out what was the time of that change. So there is some mathematical formalism involved in this information. For example, if I want to find out information such as how rats and humans are interconnected, what we can use this mathematical information, which is where n is equal to total number of substitutions, L is our number of nucleotide sites which corresponds to those substitutions. What is number of sites? Sites are basically same as websites where we have a collected information of similar things. So in nucleotide sites, we have similar nucleotide sequences which are stored together. Then k is our number of substitutions per nucleotide site. So it can be since n is our substitution and L is our site. So k is equal to number of substitution per nucleotide site. Then R is our rate of substitution, which is roughly equivalent to 0.56 into 10 raised power minus 9, and it is considered in the rate per year. So site per year is the other parameter, and it is for specified for hemoglobin alpha, which is a protein structure. Then k is equal to 0.093, so number of substitution per nucleotide sites, and this is for rat versus humans. So using this formula, if I want to find out time, it will be roughly 80 million years for rat versus humans. So when this molecular clock started, basically there were scientists Zucker, Kendall and Pauling who studied the interrelationship between different proteins, and they say that these protein structures remain constant among different lineages of organisms. So based on this information, they generalized that if this change which was between amino acids is constant, then it should be constant against all other proteins. And similarly, from this information, what we conclude that if it is constant among different molecules of proteins, then it should be constant against molecules of DNA, RNA and protein structures. So what is final conclusion is that all the mutations across macromolecules occur in the same rate in all phylogenetic tree nodes. The other one is rate of mutations. It is same for all positions along the sequences, so it will be same for all the positions. And finally, the word is that it is most suited for those species or organisms which are closely related to each other.